Hello, is it okay? Amazing Hearts, is it okay? अभी ठीक है? एक बार कमेंट करके मुझे बता दीजिए। Now it is okay? Okay, now it's okay. Okay, sorry. Extremely sorry. So today in this session, basically, let me divide the things what you have studied within 5 minutes. So today in this session, we are studying regarding the drone technology. What is drone technology? Apart from that, what is the purpose of utilizing drone technology in agriculture? ठीक है ना? So we have seen that first the question arises in your mind that why to adopt drone technology? ठीक है ना? So because you know that for the Fasal Bima Yojana, the government is employing drone technology for the purpose of assessment of the crop. ठीक है ना? Even during the time of locust attack, it was not possible for the individual to enter into the field for the purpose of spraying pesticide. So simultaneously, drones are being utilized. Looking at all these things, now the government is focusing towards the for promoting drone technology. First thing. Now, apart from that, the images that are being taken by the satellites, ठीक है ना? You know that satellites are being placed in the low Earth orbit for the purpose of assessing your resources. तो आप जो उसका भी आप जो images जो लेते हैं वो images क्या होते हैं? Blur होते हैं, ठीक है ना? Because satellites की बात करेंगे, when you talk about the satellites are being placed at a height of around 2000 kilometers, ठीक है ना? तो वो images क्या होते हैं? That is not good enough. Apart from that, the dwell time of the satellite is very low. The dwell time of the satellite is very low because if you talk about the satellite, it is parked in the low Earth orbit, and because of parking in the lower Earth orbit, it is facing great amount of gravitational forces of the Earth, and because of this क्या होता है? The satellite has to compensate the gravitational forces with the help of orbital velocity, and because of this only the orbital velocity of the satellite is very high. So because of the orbital velocity is very high, the dwell time is very low. So because of this only, now we are not relying on the satellite imagery. Rather if the images are being taken with the help of drones, they seem to be more reliable. Already we have discussed the different applications of drones. Like drones can be utilized for the purpose of irrigation monitoring. Drones can be applied for the purpose of planting. Drone technology can be applied for the purpose of spraying pesticides. Drone technology can be applied for the purpose of monitoring your crops. Even the drone technology can be applied for the purpose of soil analysis. Now, this is the concept of your precision farming. This is the concept of your precision farming. ठीक है, ये किसका है? ये precision farming का concept है. Now you know that the precision farming जो होता है, precision farming is possible with the help of drone technology and with the help of your precision farming is made possible with the help of drone technology and your nanotechnology. ठीक है ना? And your nanotechnology. Is it okay? Amazing Hearts, is it okay? Clear है? चलिए ठीक है ना? Now we'll have a little bit look regarding how this precision farming is being done. ठीक है ना? How this precision farming? So we will discuss the concept of precision farming. The concept of precision farming. Precision farming concept. ठीक है? Concept of precision farming. Concept of precision farming. ठीक है? Is it okay? चलो ठीक है. Now you know that the precision farming is possible with the help of your nanotechnology and with the help of your drone technology. So let us have a little bit idea what is precision farming. ठीक है ना? Basically in precision farming we are precisely monitoring each and every aspect of the agricultural thing. Now how it is being done? Suppose this is agricultural plot. ठीक है आपका? This is agricultural plot. Okay. This is your agricultural plot. Now, basically we are utilizing nano-based sensors. What is the importance of the precision farming? The precision farming जो है, मैंने बताया nanotechnology and drone technology. So here what you are using this nano sensor. What is this? This is your nano sensor. Nano sensors. Why you are using nano sensors? Nano sensors are being utilized because its accuracy जो है, it is greater than 99.9%. The accuracy of nano sensor is greater than 99.9%. What is the reason behind it? The reason is सरफेस एरिया टू वॉल्यूम रेशियो इज वेरी हाई, सरफेस एरिया टू वॉल्यूम रेशियो जो होता है इसका बहुत हाई होता है। रीजन इज सरफेस एरिया टू वॉल्यूम रेशियो इज वेरी हाई, सरफेस एरिया टू वॉल्यूम रेशियो जो होता है वो काफी हाई होता है, ठीक है ना। अपार्ट फ्रॉम दैट वी आर हैविंग द स्प्रिंकलर अरेंजमेंट, दिस इज व्हाट, दिस इज योर स्प्रिंकलर अरेंजमेंट, this particularly nano sensor, it is being utilized simultaneously for the purpose of monitoring your fertility of the soil, for the purpose of monitoring your soil moisture, even for the purpose of monitoring pest and weedicide, ठीक है ना। apart from this, this is the drone, ये क्या है आपका, this is your drone, okay, and all these things are being controlled by this computer, ठीक है ना। now what happens, suppose your nano sensor senses that the moisture content of the soil is declining, so what it will do, the same, the nano sensor is also connected to the computer. Now it will send a signal to the computer that the moisture content of the soil is being declining. Now this computer, it will give the command to the sprinkler system. The sprinkler system, it will start spraying the water, and after sufficient water is being, after your plot is being irrigated, again the nano sensor will check, yes, now sufficient water is available. It will give a feedback command to the computer. Now sprinkler system will stop. Same, if your nano sensor is monitoring suppose fertility of the soil is being dropped, so what will happen, it will give a command, a computer system will give a command to the drones, it will start spraying fertilizers. If your agricultural plot is being infested with pest, again nano sensor will sense it and the computer will give a feedback to the drone, and drone will start pesticides. So this is a little bit concept regarding the precision farming. This is all about the precision farming. Is it okay? So we have completed your drone technology. Now we have to see benefits. So what are the benefits of drone technology? Already we have discussed each and everything. So what are the benefits of your drone technology? Benefits of drone technology, what is it? So you can see that enhanced production, that means if drone technology are being utilized, more or less we are monitoring each and every aspect of the soil, that means more or less precision farming concept, we are monitoring the things precisely, and because of this the productivity of your crops can be enhanced. Greater safety of the farmers, suppose if you are spraying chemicals with your hand, no doubt you are using the muscle, but you are not 100% safe. Rather, if you are spraying chemicals with the help of the drones, it is more safer. Faster data for quick decision making. Obviously, with the help of the drone, you can easily monitor your complete agricultural field in different aspects. Soil analysis can be done, pesticides, weedicides can be checked, your fertility can be also be ensured, productivity can be ensured. तो जितना जल्दी आपको पता चलेगा, you will ensure different steps for the purpose of increasing its performance. Less wastage of resources, useful for insurance claim. ठीक है, चलिए. Now the most important thing is, no doubt we want to employ drone technology, but when you talk about it is not being utilized, why? What is the reason behind it? Farmers are not employing drone technology. Yes, if you talk about Punjab region, if you talk about Haryana region, ठीक है ना, if you talk about वेस्टर्न यूपी रीजन, वहां पे अगर हम बात करेंगे वेयर एग्रीकल्चरल प्लॉट जो होते हैं काफी बड़े होते हैं, वहां पे यू कैन एम्प्लॉय ड्रोन टेक्नोलॉजी, बट इट इज नॉट पॉसिबल एवरीवेयर. ये आपका हर एक जगह जो है पॉसिबल नहीं है. व्हाट इज द रीजन? बिहाइंड इट? यू कैन सी दैट अराउंड unviable commercial operation due to small scattered land holding. India में क्या, we are having a small scattered land holding. If you see 86% of the farmer in India, they have land holding less than 5 acres. So one of the important aspects, जनरली आप ड्रोन्स की बात करेंगे, परफॉर्मेंस की बात करेंगे, तो वहां पे if you are having land of greater than 100 acres, तो वहां पे आप ड्रोन टेक्नोलॉजी can be utilized for the purpose of assessing, monitoring, है ना, even for the purpose of spraying seeds, pesticides, fertilizers. But 86% of farmers in India जो है, they are having less than 5 acres. तो वहां पे ड्रोन टेक्नोलॉजी का इस्तेमाल कर ही नहीं सकते. यू कैन डू इट, लेकिन आप नहीं कर सकते बिकॉज आपको ड्रोन परचेज करना पड़ेगा, ठीक है ना, एंड इट सीम्स टू बी वेरी कॉस्टली. लिमिटेड फ्लाइट टाइम रेंज ऑफ ड्रोन्स ड्यू टू रिलेटिवली हाई पेलोड 20 to 6 मिनट. तो आप देख सकते हैं कि इफ यू टॉक अबाउट द flight time is very less, that is equivalent to your 60 more, मतलब 20 to 60 minutes, 20 minutes to 1 hour. High initial cost of drones with maintenance issues. Connectivity is in rural area for online coverage. Now, what is the problem in rural area? The problem क्या वहाँ पे? Problem ये है, if you want to monitor your agricultural plot, for the purpose you need to have a connectivity, ठीक है ना? लेकिन वहाँ पे क्या connectivity is missing? Connectivity is missing. Knowledge and skill issues, as it needs specialized skill, knowledge from farmers to utilize drone input, concerns over misuse of drones to infringe privacy and security, ठीक है ना? That means even the drones can be utilized for the purpose of for the violating privacy. ठीक है, किसी के घर में घुस गया, वहाँ पे चीजें ऊपर से मॉनिटर कर रहे हैं. You can understand the drones जो it can also be utilized for the purpose of violating privacy. Okay. Now this is okay. Now we have to see self-amplifying messenger DNA, self-amplifying messenger RNA vaccine. So you can see that self-amplifying messenger RNA vaccine show promising result against COVID-19. So earlier we have utilized messenger RNA, or earlier we have utilized messenger RNA vaccine. You know that Pfizer and Moderna, they are being prepared with the help of messenger RNA concept. But now we are moving toward the self-amplifying messenger. So now let us have an idea regarding how this messenger RNA vaccine works. How this messenger RNA vaccine works. Okay? So firstly we will know. So how this messenger RNA vaccine works. For this purpose we have to see the central dogma of molecular biology. For this purpose we have to see the central dogma of molecular biology. mRNA vaccine working. mRNA vaccine working. mRNA vaccine. ठीक है ना? So to understand mRNA vaccine, we have to look for central dogma of molecular biology. क्या? Central dogma of molecular biology. तो क्या होगा central dogma of molecular biology? Suppose this is your cell. ठीक है ना? Inside the cell we are having nucleus. Inside the nucleus we are having chromosomes. क्या इसके अंदर में क्या होता है? We are having chromosomes, and inside the chromosomes what? We are having DNA. है ना? We are having DNA. So suppose this is your DNA. This is your DNA strand. Now what is there in the DNA? Genetic informations are there. Genetic informations are there in the DNA. Now what will happen? यहाँ पे क्या होता है? RNA polymerase enzyme is there. What? RNA polymerase enzyme. What is the function of RNA polymerase enzyme? Its function is to copy all the genetic informations of copy all the genetic information from the DNA and paste it into the messenger RNA. That means what RNA polymerase do? RNA polymerase is using your DNA as a template, ठीक है ना? And the genetic information from the DNA is copied and finally it is pasted to the messenger RNA. Now this messenger RNA is prepared. Now this all the genetic information जो है is copied and it is finally pasted into the messenger RNA. This is your messenger RNA, and in more technical language we'll call it as your transcriptome. क्या कहते हैं हम लोग? Transcriptome कहते हैं. Transcriptome कहते हैं. Kryptome कहते हैं. So this process, the process by which the genetic information of your DNA is being copied and pasted into the messenger RNA, all this process is called transcription. इस process को क्या कहते हैं हम लोग? We call this process as transcription. इस process को हम क्या कहते हैं? Transcription कहते हैं. ठीक है ना? Transcription कहते हैं. Now what happened? Whatever we have discussed, all this thing is happening inside the nucleus. So if you analyze the nucleus, we are having nucleopores. Holes are there. These are having the nucleopores. Now what will happen? This messenger RNA, in which the genetic information of the DNA are being copied, this messenger RNA will move out from the nucleus through this nucleopores. Now where it will go? It will go to the ribosome. ये क्या आपका ribosome? This is called your ribosome. ठीक है ना? And ribosome इसको हम लोग क्या बोलते हैं? Site for protein synthesis. We call it as site for क्या बोलते हैं? Site for protein synthesis, because basically ribosome is responsible for synthesis of the protein. So this mRNA will move closer to the RNA, move closer to the ribosome. Now what the ribosome will do? Ribosome के अंदर में दो पार्ट होता है. One is a smaller subunit and other is a larger subunit. what is the function of a smaller subunit? The function of a smaller subunit is to understand the genetic information, that is there in the messenger RNA. And once the genetic information is being decoded, the same is being transferred to the larger subunit. And what is the function of the larger subunit? The function is to, whatever the genetic information it is there, it will prepare the protein accordingly. Suppose यहाँ पे when you talk about हमारा यहाँ पे genetic information of our क्या hair style, genetic information of hair क्या है, genetic information of hair, ठीक है ना. So now you can see that the protein यहाँ पे बनेगा, वो क्या बनेगा, वो आपका hair बनेगा. क्या बनेगा, hair बनेगा. And it is called alpha keratin. क्या होता है, तो प्रोटीन क्या होता है, alpha keratin. So this process is called translation. Translation means the genetic information is being translated to the respective protein. So this process, the process by which the genetic information that is being there in the messenger RNA is being converted into the respective protein, this process is called translation. इस process में क्या होता है, translation कहते हैं. क्या कहते हैं, translation कहते हैं. Now we will see how this messenger RNA vaccines work. Is it okay? Is it clear? बताइए. Is it clear? जल्दी से बताएं. If any doubt, please do let me know. Now we will see how Pfizer and Moderna vaccines are being prepared with the help of this technology itself. ठीक है, चलिए. So now we will see how the Pfizer and Moderna vaccines जो होता है, they are being prepared with the help of messenger RNA. Because once we understand the messenger RNA, then only we can see how self-amplifying messenger RNA vaccine works. ठीक है ना, how self-amplifying messenger RNA vaccine works. So you can see that Pfizer and Moderna जो होता है, they are being prepared with the help of your messenger RNA. Pfizer and Moderna, ठीक है ना, Pfizer vaccine. Pfizer and Moderna. Pfizer and Moderna, ठीक है. They are being prepared with the help of your messenger. They are being prepared with the help of your messenger RNA vaccine, ठीक है ना, messenger RNA. Now, how these things are being prepared? You are quite aware that how coronavirus attacks our body. Let me give you 1 minute. ठीक है ना. This is the structure. This is the coronavirus. Inside the coronavirus, what is there? The genetic material was your RNA. What is the genetic material of coronavirus? It is your RNA, and that is single-stranded RNA. Because if it was DNA, तो उतना ज़्यादा mutations आपको देखने को नहीं मिलता. DNA is little bit stable in nature. But when you talk about the genetic material of RNA, when you talk about the genetic material of coronavirus, it is single-stranded RNA, and because of single-stranded RNA, it is having the higher probability of mutation, and that is why the coronavirus is mutated too. Now, over the surface, these spike proteins are there. Over the surface, these spike proteins are there, and spike proteins are there. And in our cell, suppose this is our cell, when you consider our heart cell, kidney cell, lung cell, a special type of enzymes is present over the surface. This is called angiotensinogen converting enzyme. ठीक है ना? Over the surface, we are having a special type of enzyme. It is called ACE2 enzyme. ACE2 enzyme. That is called angiotensinogen converting enzyme. Now, this is spike protein and ACE2. They are having greater bonding. बहुत ज़्यादा इन दोनों में प्यार मोहब्बत है. They are having a greater bonding. They are having a greater affinity, and because of this, the spike protein penetrates into your cell through the gateway of ACE2. Are you getting my point? Over the surface of a coronavirus, spike proteins are being present, and when you talk about certain body cells like heart cell, lung cell, kidney cell, over the surface ACE2 enzymes are being present, and a spike protein of the coronavirus and ACE2 enzyme, they are having greater binding affinity, and because of this binding affinity, the coronavirus penetrates into our cell through the gateway of ACE2. So now, who is the main culprit? Now it is a spike protein. ठीक है ना? Now, so you know that we have already conducted the genome sequencing of coronavirus are being done. Genome sequencing is being done. So when we do the genome sequencing, we are quite aware of how spike protein is being made, what are the genetic information of spike protein. So now how this mRNA is being prepared. Suppose this is your mRNA strand. In the lab, you have prepared mRNA strand. Okay, and from the lab, itself, we have prepared the genetic information of spike protein. This is what genetic information of spike protein. This is called genetic information of spike protein. Genetic information of a spike protein, and it is being prepared in the lab. ठीक है ना. So this is your mRNA. Now it has been converted into your respective vaccine, and once the vaccination is given, now this is since this is an mRNA, since what this is an mRNA, it will go to the ribosome. ये कहाँ जाएगा? It will go to ribosomes. ठीक है, it will go to ribosomes because it is the mRNA. Definitely. Now what will happen? Now, so you have seen that this particular mRNA, when it enters into your cell, it will directly go to the ribosome. And what is the purpose of ribosome? We have just discussed the purpose of ribosome is to synthesize the respective protein from the genetic information. Now what will happen? What is the genetic information? The genetic information is spike protein. So the ribosome will study the genetic information, and the same genetic information will get converted into the respective protein. So what will happen? It will produce the respective protein. This is called spike protein. This is called spike protein. The spike protein is being it is being prepared in our body with the help of your mRNA vaccine. Now think of this. This particular spike protein जो हमारे बॉडी के सेल ने कमांड नहीं दिया इसको बनने के लिए, it has been extremely feeded. जब आपको वैक्सीनेट किया गया तो वैक्सीनेशन में आपको mRNA स्ट्रैंड दिया गया, एंड जो mRNA स्ट्रैंड जो इसके अंदर में जेनेटिक इंफॉर्मेशन किसका था? इसके अंदर जेनेटिक इंफॉर्मेशन आपका स्पाइक प्रोटीन था, और उसी के कारण आपका स्पाइक प्रोटीन बना हुआ. नाउ इट इज योर एंटीजन. इट अपेयर्स टू बी फॉरेन पार्टिकल फॉर बॉडी. ये क्या हमारे बॉडी के लिए फॉरेन पार्टिकल है. दिस इज योर फॉरेन पार्टिकल फॉर द बॉडी. नाउ इट अपेयर्स टू बी एंटीजन फॉर आवर बॉडी. एंटीजन for body. So, and because of this, now our immune system will activate. Immune system में B lymphocytic cell जो है, this is a B lymphocytic cell. It will get activated, and it will release antibody. Okay, against this spike protein. But something now, ये आपको तो पता है कि कोरोना वायरस तो है नहीं completely. कोरोना वायरस ना dangerous है, because what is a virus? The virus basically it hijacks our own machinery, and it will start replicating. लेकिन यहाँ पे complete virus is not there. Only just a spike protein is there. ठीक है ना? And ये spike protein आपके लिए harmful नहीं है. But since it is a spike protein, antigen, foreign particle, our immune system will get stimulated, activated, and it will release antibody. Now these antibodies are being released. But since our immune system has been activated, immunological memory will get developed. What is it? Immunological memory जो होगा, immunological memory will get developed. Now, what is the purpose of immunological memory? Lymphocytic cell immunological memory remembers that I have released antibody against a particular antigen. Are you getting my point? It basically remembers that it has released antibody against a particular antigen. It retains the information. It retains the information that it has released antibody. It has released antibody against a particular antigen. Okay, antigen. So now what happens? Suppose this is the original coronavirus. Now this is the original coronavirus. ठीक है ना? This is the original coronavirus. When it enters into your body, penetrates into your body, from the immunological memory, B lymphocytic cell tries to remember कि यार whether this virus is a new virus or I have released certain antibody against it. So it tries to put pressure on the immunological memory. भाई सोच के बताओ कि यार हमने इसके खिलाफ एंटीबॉडी रिलीज किया, और फिर इम्यूनोलॉजिकल मेमोरी जो होता है वो बताता है yes we have released antibody against this particular spike protein. Spike protein ही है क्योंकि only spike protein. तो ये क्या करता है, झट से आपका क्या करता है, एंटीबॉडी रिलीज हो जाता है और आपका वायरस जो है न्यूट्रलाइज हो जाता है. ठीक है ना? तो दिस इज हाउ लॉन्ग लास्टिंग इम्यूनिटी इज बीइंग क्रिएटेड, ठीक है ना. So this is what we have discussed. We have discussed the mRNA technology. mRNA technology. One thing to be remembered here: only translation process, transcription process is not happening in mRNA vaccine technology. Only translation happens. याद रखिएगा. यहाँ पे mRNA technology की अगर हम बात करेंगे, when we talk about mRNA technology, in mRNA technology, ट्रांसक्रिप्शन जो होता है, ट्रांसक्रिप्शन डू नॉट टेक प्लेस only translation happen, only translation happens, ठीक है ना. Translation happens. Okay, this is okay. So we have seen how mRNA technology works. Now we have to see what is self-amplifying mRNA. Is it clear to everybody? Please do let me know. Is it clear? बताइए, is it clear? So now we have to see what are the advantages of mRNA technology. First of all, when you talk about the mRNA technology, it is very much economical in nature. First thing, remember, we're talking about the advantages. If you want to write it down, you kindly write it down. So first of all, it is very much economical in nature. Why economical? Why economical? Because here we are not culturing pathogen. Remember, we are not culturing pathogen. जैसे हम अगर बात करेंगे, when you talk about the polio vaccine, ठीक है ना, पोलियो में there is two way. Either you go for the live attenuated vaccine. Live attenuated means the polio virus को हम लोग kill कर देंगे, genetic materials are being destroyed, ठीक है ना, वो एक खोखा बन जाएगा. We are utilizing it. So that is called live attenuated vaccine. Sorry, live attenuated vaccine में क्या करते हैं हम लोग, वायरस को कमजोर कर देते हैं, genetic material destroy कर देते हैं, weak कर देते हैं. And the other is your killed vaccine, ठीक है ना. other में हम लोग क्या करते हैं, हम लोग inactivated vaccine, वहां पे हम लोग killed version of the virus इस्तेमाल करते हैं. In both of this, what we are doing basically utilizing the virus. So for that purpose we have to go for the culturing of virus. यहाँ पे आप देख सकते हैं, here we are not using any virus, rather we are utilizing the genetic information of the spike protein. So first is it is very much economical in nature, and second, the culturing of the virus is not required. Third thing, what you must remember, you must have seen the vaccine derived polio virus. Vaccine derived polio virus. It is a case when we are going for the live attenuated vaccine. Generally, what happens in live attenuated vaccine, we are using the live virus, but it is in the weakened form. Sometimes, and out of 38 lakh, only one cases happens, that virus will get mutated. And it will become very much severe in nature. So that is called vaccine derived polio virus. So it happens in the ratio is around 38 lakh to 1 people. That means out of 38 lakh people, only one such type of mutation will happen. But since your virus are not being utilized, we are utilizing the genetic information only. So there is no such type of cases. ऐसा cases आपको देखने को भी नहीं मिलेंगे. So these are the advantages. These are the advantages of this technology. Now what we will see? We will see what is your self-amplifying mRNA. What is self-amplifying mRNA and how it is being different from your messenger RNA? First of all, you must know in messenger RNA we have utilized the genetic information spike protein. That means only one type of protein is being utilized. Are you getting my point? In mRNA, messenger RNA technology, we have utilized only one type of protein. एक ही type of protein का इस्तेमाल किया गया. कैसे? In messenger RNA, you have seen that we have in messenger RNA we have embedded only the genetic information of a spike protein, and so only one type of protein is being created in your body by the process of translation. एक ही, तो वो क्या आपका सिर्फ एक ही type of protein का इस्तेमाल कर रहे हैं. But in self amplifying mRNA हम लोग क्या करते हैं? A self-amplifying mRNA vaccine is an important over the traditional mRNA as it encodes 4 extra proteins. So आप देख सकते हैं 4 extra proteins is being encoded. When you talk about the mRNA, the only protein that is being produced is mRNA में क्या होगा? When you talk about mRNA, just we have seen, ठीक है ना? We are particularly dealing with only one type of protein, that is the spike protein, ठीक है ना? Spike protein क्या? Spike protein, ठीक है ना? But अगर आप बात करते हैं सेल्फ एम्प्लीफाइंग mRNA, तो हियर जो है यहाँ पे जो है it encodes 4 extra protein. क्या यहाँ पे it is encoding 4 extra protein in addition to the vaccine antigen, in addition to spike protein. तो आप देख सकते हैं if we are sending in the mRNA, if we are attaching genetic information to spike protein, definitely by the process of translation spike protein will get created. It seems to be a foreign particle antigen. वो आपका फिर क्या होगा, वो आपका क्या करेगा, उसका एंटीबॉडी रिलीज करेगा. बट व्हेन यू टॉक अबाउट द सेल्फ एम्प्लीफाइंग mRNA, apart from spike protein, 4 extra proteins are being fed. तो आपको क्या होगा, 5 in our body, 5 different type of protein will be there. One will be a spike protein and 4 different type of protein will be there. अब वो कितना टोटल हो गया, 5 different types of proteins हो गया बॉडी में. तो मतलब क्या हो गया, 5 different types of antigen. So हमारा immune system जो है वो क्या करेगा, it will release antigen against all of them. It will release antigen against spike protein apart from the other protein जिसको आपने सेंड किया by the process of self amplifying mRNA. तो 5 different type of antibodies will be produced. So आप सोच सकते हैं कितना अच्छा आपका immune response होगा. तो this is what your self amplifying messenger RNA. ठीक है. Now what are the advantages? Advantages of self amplifying mRNA vaccine over the conventional mRNA vaccine is making storage easy, minimizing dose of mRNA, lowering the cost of vaccine. Because here the immune response की बात करें, immune response is good because apart from the spike protein itself, we are sending 4 different types of proteins. So total जो है our immune system will get stimulated, activated, and we will try to release 5 different type of antibodies. 5 different type of antibodies. ठीक है. Advantages of already we have seen. So this is what you now this is a mRNA and this is a self amplifying mRNA. ठीक है ना, यहाँ पे कितना 5 different types of 4 different types of proteins are being produced. Is it okay? Clear? So today we have taken only 2 topics that is most important from the perspective of mains. The next time we will take another 2 topics. ठीक है ना? If any doubt, please do let me know. If even those students who are watching my lecture in offline mode, if any doubt, please do comment in the comment box. ठीक है ना? So thank you, thanks a lot everyone. Jai Hind, Jai Bharat.